Department of Chemistry, Faculty of Science, Assays University, will be giving his talk on the application of computational design, synthesis, and biological evaluation to target diseases of the developing world. So please give Dr. Gleeson a very big hand, please.
So thank you very much, Ajahn Tannen. Thanks to the committee, Jarl Tannen, for the kind invite. Sorry for forcing you to come in so early when our speaker wasn't ready. I'll try to keep the time, but I think I've got far too much, so I guess we've got time after for more poster analysis. So the talk I'd like to give today is it's ongoing work that I've been doing with Thai collaborators to, well, really continue what I did in Europe when I was working within the pharmaceutical industry. So I was involved in the computational design aspect with colleagues in chemistry working on synthesis and colleagues in biology, high throughput screening, working on the evaluation of compounds. So our goal, well, I've been here 7 years now, and the goal over that time is to try to, well, envelop myself in a environment maybe that I had in the pharmaceutical industry in the past, or at least have the capability to do some aspects of the research. So I'll discuss some of the things that we've been doing to try and generate some degree of well, I won't say drug discovery capability, but maybe probe hit generation work that we've been doing. So in the talk, I'm going to talk about the use of, well, Chem bioinformatics approaches that we've used to try and reduce the complexity of our research. We've got a limited number of students, limited budget, very limited budget compared to a lot of the researchers here. So we try to use these methods to understand, well, we have a disease focus first, and we try to bias the selection of our compounds or the area that we work in to particular targets. We think about what kind of compounds we want to interact with the target to affect the disease. We also think really what synthetic routes do we want to develop to synthesize the compounds to hit the target to affect the disease also, and then what kind of properties that we want to predict or we want to obtain. So really what we go on is really looking through the wealth of information that a number of speakers have talked about, and Hersey, Tudor, Opera as well in detail, how we can select or bias the selection of compounds for synthesis or screening, and then putting them into phenotypic assays. So I'll discuss a little bit about the computational design, the synthesis and the phenotypic screening that we've been involved in. So my group's interest over the last few years really has been computational chemistry and chem informatics, sort of bioinformatics. But the kind of the capability that we want to do is more chemical biology. Really like some of the earlier speakers, Roman's talk, for example, not as, we wouldn't aim to, I mean, trying to understand targets in terms of chemical biology. So in our group we wouldn't be able to do that. We've got the chem informatics, we're in the process of developing and strengthening our synthetic ability, and we get phenotypic assays with collaborators in biology, microbiology around Thailand. So we have identified a number of very capable researchers who are doing detailed mechanistic studies that we can get our compounds tested in. So the hope is really to try and do some interesting chemical biology. I didn't put drug discovery, drug design, drug lead generation. Really we're interested in really trying to understand more carefully the disease target link or compound disease target link. Okay, so our real focus is on diseases of the developing world, and that's pragmatic primarily because we can get funding from Thai granting agencies, and our collaborators can also get funding from Thai granting agencies, and they've assays already set up, so they don't have to develop, we don't have to set out to develop the biology capability ourselves, we can harness what's already developed within the country. And these are low to medium throughput phenotypic assays, so malaria, dengue, cancer, bacteria, fungi, and some of the posters you would have seen outside discuss or mention some of these capabilities. So from a chemistry point of view, I'm not a trained organic chemist, so we really have to focus on chemotypes with tractable synthetic roots and good yields, and that involves quite a lot of data mining, informatics, text searching, literature searching, patent searching. So we need reagents, well, we need roots that are quite reliable. We need good reagent availability, and we need reasonable cost. So it's a lot of computational searching, analysis, and to try and work out the most optimal set of compounds. And so we also focus on chemotypes that have the potential to be modified to other targets or diseases. In that way, if we invest, so this research has been done by MSc and PhD students at the university who don't necessarily have a strong background in organic chemistry. So when they've developed the understanding of the root, we want to synthesize a number of different compounds to cover potentially a number of different targets, so that we can build a biased sort of library so that we can use in the future. So it's not simply about targeting the disease, it's also about generating a sort of a focused library on targets that we know are implicated in diseases that we can get screened within Thailand. And so that really led us to the focus of kinase inhibitors, which was something that I worked on quite a lot in industry. So I'm sure a lot of you know better than I. They're involved, well, they're critical in cell signaling and proliferation, differentiation, migration, apoptosis. They've been extensively studied for targets such as cancer and inflammation. And there's approximately 30 marketed kinase inhibitors to date. And there's extensive information that they can also be used to treat other diseases. So for example, I've just collected a few papers, but basically cancer inflammation, that's well established, well known. Kinase inhibitors are also known to work against malaria. I have a few more papers. This is just an example of one. Also potentially these JAK kinase inhibitors are known to affect replication in HIV infected cells. ERK inhibitors functioning to reduce dengue virus injury, JAK-STAT in Alzheimer's and Huntington's disease. So there is potential for using kinase targeted inhibitors at well-known cancer targets, and inflammation targets for other uses. So just to highlight that, for example, if we take 4-phenyl, so we've got a pyrimidine here, I'm not sure if it's clear, but at the 4 position we've got a CC bond with a phenyl or other aromatic ring, and at the 2 position we've got an aromatic amine. So these compounds are relatively easy to generate, Suzuki coupling here, acid base or palladium coupling at this point. And with that type of chemistry you can generate JAK kinase templates, which literature, patents and papers show work for cancer. Here we've got data that shows that they function on antibacterial pathways, so there is an antibacterial kinome also. These are able kinase inhibitors. We've got MAP or GSK or GSK3 kinase inhibitors that also have antifungal properties. Other CDK-like inhibitors with antifungal activity and IKK compounds. So by focusing on particular, say for example, JAK kinase inhibitors, we can enumerate further structures that could potentially be useful in a library that target other kinases. So the goal is really to select compounds that are interested in particular diseases or targets, and then while having invested in the root, synthesize further analogues to try and build up sort of the diversity of chemotypes that we have within the group. So after extensive literature searching and analysis and looking through patents and papers, you can come up with ways to modify. So starting with 2,4-dichloropyrimidines, we can end up with, at the 4 position, we can end up with O-linked, CC-linked, or NH-linked. We can use Buchwald-Hardwick amination. If for difficult cases, we can use acid-base catalysis, or we can use base catalysis to add the amine at this position. So using the same reagents and the same methods, we can generate relatively straightforwardly quite a large number of chemotypes. So as I said, we focus on targets and diseases important to Thailand. The libraries have been synthesized by MSc and PhD students, and these are students not necessarily from an organic chemistry background, so what they're doing, I think, is exceptional work. So we've taken a focus on sort of MAP kinases and tyrosine kinase, and we are kind of target agnostic. We don't really care what, well, at this point we're not necessarily worried what target we're hitting. We're interested in generating interesting compounds that we can use as probes to try and understand a phenotypic response, and with colleagues we can then look in more detail at the potential pathways that are operating, or the hits that we find. We can infer a potential target from cross-referencing with literature sources, or looking for the most equivalent human kinase, and then maybe if it's in a bacterial, fungal, or parasite, we can then use bioinformatics approaches to identify the most likely candidate in the species that it's functioning via. So the goals that we have are really novel probes or hits, and I don't really think to generate a lead requires quite a lot of additional testing and evaluation. So I mean, we're focused on hits, novel probes, and really scientific publications, and just providing an education for the students that are in the faculty, the department that I'm in. So to do the chemistry, we really rely on extensive patent searches and literature searches, and we try to then introduce novel design elements using structure-based design or ligand-based design to try and move beyond maybe the patent scope of the original compounds that we've identified. Although in many cases the patent might be for cancer, so using the compounds for another indication isn't necessarily an issue. Not that we're necessarily interested in taking compounds further, at least at this stage. So we focus on tractable synthetic chemistry and focus on molecular properties with reasonable molecular properties, try to keep molecular weights and log P's low. We're focusing on phenotypic endpoints, so there is a degree of added solubility, permeability in the assay, in the sense that compounds that are very insoluble aren't going to show a reasonable activity. So to some extent there is using a phenotypic approach helps mitigate against very, very undesirable physical properties. So we had to build up we're building up a larger library, but we've got maybe 150 diverse amines, halides, alcohols, 40 or so boronic acids and other building blocks and templates, so that there's sufficient diversity in that we can now generate quite a number of novel compounds relatively quickly. And then this is not very detailed computational chemistry, but for non-computational chemists it can sometimes be useful. Basically what we use very basic ligand structure-based methods. So for example, PDE5 inhibitor, we want to design a molecule that if we find a hit that's similar, we want to design a new molecule. We make a structural modification. When we compare the similarity between the molecules, we find it's high, but it lies outside the original scope of the patent, or so we basically scaffold hopped. Or if we've got a protein structure, we can look at the active site, characterize the binding sites, and try to modify the structure roughly. So it's not necessarily detailed computational chemistry that we're doing. A lot of it's dictated by the reagents that we have, the reagents that will work for the route that we're using also. So there's an element of practicality in the computational chemistry that we do. So if we're interested in a particular disease, for example, we'll start off with patent or literature searching, look at phenotypic data, try to look for chemotypes, assess the synthetic tractability, design some modifications or synthesize some analogs of known compounds just to try and understand the SAR or the possible targets. Synthesize, characterize the structures, then do biological testing, do some standard SAR, optimize the compounds. So we focus again, HIV, malaria, dengue, and again we try to focus on designs outside of known target space, but if we're working within a patent that someone else has generated for cancer and we're interested in looking at malaria, it's not that much of an issue really. So current screening, so we have got collaborators with where we can do malarial, bacterial screening, so multiple bacteria strains. We've also established collaboration with people doing dengue, fungi, cancer in diseased patients or TIE-focused or prevalent cancers, for example, Alzheimer's disease. And then there's also possibility to look at influenza and type 2 assays, so HIV-1RT and virus equivalent are considered quite an issue in Southeast Asia. So at the moment, we don't have a huge amount of compounds. There's 120, but maybe there's 15 distinct subtypes in there. So synthesized by 4 MSc, PhD students in 18 months, and we could add 20 compounds to that every month, I think. So what we've done is we've clustered the data based on input from known targets that might be important, or sub-selected certain compounds for screening, tried to make it diverse to some degree. And we've tested it in these 2. We've submitted it to these other 4, but we have found some malaria hits and some bacterial hits that we're currently in the process of trying to work up. And so that involves synthesis of further analogs and profiling in other assays of interest. So I just mentioned a bit about the anti-malarial lead generation. So I think Tudor and Anne mentioned a lot of the open access data released by companies recently, over the last few years. So it's a great source of data for academics working in this area, a neglected area where industry doesn't really want to work anymore. So looking at malaria, we can see that malaria, STAT6, protein kinase parasites from Dehrig has done a lot of work on this, CDK-like kinase involved in parasite. And last year, actually about a year into the plans that we had to do this, a paper by Emily Derbyshire, chemical interrogation of the kinome came out, and they had taken a subset of 1,000 known kinase inhibitors and tested them in Plasmodium falciparum assay and came up with, so it was a kinase focused library, and came up with 30 or so hits for multiple kinase. So for example, we've got CDK MAP, so MAP kinase, GSK MAP kinase, and then Aurora, VGFR. So there's some JAK kinases in here, JUNK, MAP kinase. So there does seem to be certain distinct classes of kinase compounds and particular human kinases that appear to be important. So it's not clear whether kinase inhibitors function via the human kinome or the malaria kinome, but there are known similar malaria kinases to CDK and to Aurora JAK, for example, which suggested maybe working via either. So in this case, we selected kinase targets that were implicated in malaria based on both the Derbyshire paper and earlier papers, so trying to bias the selection of compounds to chemotypes that are likely to inhibit the human kinases that have been identified by others. And then we went through the screening data, the HDS screening data, and identified kinase-like compounds that fit the criteria of, they look like kinase inhibitors, and we took those forward also. So two ways of doing it. So the GSK paper of Gamo in 2010, they reported a number of different inhibitors, but we had maybe a look in through the data and saw these inhibitors. So they're like Aurora JAK2 inhibitors based on analysis of the patent literature. And these have also been sort of very similar to what the Derbyshire paper showed, Aurora JAK tyrosine type kinase inhibition. They show pretty good activity. And then there's some cytotoxicity, but it's not unexpected for cancer compounds. But the selectivity of 10 to 50-fold, which is generally specified, isn't necessarily a problem with these. At least the SAR that we've looked at suggests it is possible to remove cytotoxicity. So if we look through the patent literature, we can see, for example, JAK, Aurora JAK, JUNK kinase, JAK2, and these are all for cancer indications. So the targets are quite well known. They come up as interesting hits in high throughput screening. The targets have been implicated by others previously, so there seems to be, reasonable confidence in them. One issue though is when you look through the patent literature, you do notice that, like most kinase inhibitors, the selectivity is a function of these kinase inhibitors, these ATP binding site inhibitors. So they do inhibit a number of different targets at above 50%. I think this is a 10 micromolar inhibition. So it does have a kinase profile. But then there's also this paper. So originally when this was published in, I think it was 2012, I think this is one of the MMV compounds. It showed extremely good efficacy in a Plasmodium bergii model, but they didn't really know the molecular target. They thought it was kinase, and they suggested maybe we could profile against a human kinase panel to try and understand, or Plasmodium panel. But I think it turned out that it inhibited, or recently came about that it inhibits a PI3 or PI4K kinase, or not tyrosine kinase, even though that was originally what it was thought. So when we talk about the kinase activity or the potential target, I'm not necessarily sure exactly what it means, or exactly which targets, or which the precise profile, what it means exactly. If it works in the phenotypic assay, it doesn't have side effects. Potentially that's okay. So if we look at, how are we for time? Okay, so looking through the hits that we have, so these came up as Aurora JAK kinase hits from the HTS, and looking at the sequence similarity between the malaria kinoma and these targets, you can find PFPK7, which has 23% sequence identity with Aurora, 16% JAK, but about 40% sequence similarity. So these two targets coming up is again consistent with the screening data reported by Derbyshire. And PFPK7, it's expressed in several growth stages in the mosquito vector and human host, and it is believed chemical inhibition should slow the parasite growth. Interestingly, so if you look at the diaminopyrimidines in the PDB, so if we were to synthesize further analogues of these, it wouldn't necessarily be a major difficulty to synthesize additional compounds for CDK, JUNC, ERK, LCK. So that, well, these are compounds that are bound to crystal structures of the protein, which suggests that we could generate a diversity of structures relatively easy by undertaking a project to synthesize these for malaria also. So we initially synthesized 19 analogues around the series identified from screening, and some of these overlap with the patent space of the patents I've discussed, and others lie outside. And so we've tested the compounds it's a tie-resistant strain, so the K1 strain that is located in Southeast, or prevalent in Southeast Asia, and we also assessed the cytotoxicity in viral cell line. So this is a work in progress, but from what we could see, we found a number of compounds did have confirmed activity. Cytotoxicity at, I think we measured a 50 micromolar was noticeable for some compounds, but there was a window, and 10 to 50 fold should be achievable. Interestingly, the original results reported by GSK gave PXE50s of around about 8, whereas we get maybe 6.5. But the best compounds are sort of not necessarily so far off mefloquine, which is one of the standard treatments for malaria. So it works via a different mode of action also, which is potentially a benefit of employing kinase inhibitors to treat malaria as a way to try and defeat disease resistance. And so at the moment we're testing in representative human kinases to try and understand the difference between these. Testing in the malaria kinome is not necessarily trivial, but that's something that we're considering. And we're also testing in other Plasmodium falciparum strains to see whether the results are generalizable or not. So in conclusions, we have used computational and synthetic methods to produce a small scale library biased towards targets or chemotypes implicated in diseases of the developing world, so dengue, malaria. So we've made extensive uses of bioinformatics and cheminformatics tools to try and help us bias the selection synthesis of compounds or targets that we focus on. Some early screening results that we've obtained in malaria and bacteria suggest a hit rate of around 10% for these, which isn't necessarily too bad. These are all roughly, say, 1 microgram per mil in bacteria, less than, or around about 1 micromolar in Plasmodium falciparum. So they appear to be reasonable probes in the K1 strain, and we've added further analogues into the kinase library that we're trying to develop, we can again, through different iterations, submit more and more compounds and try to generate more and more information. So it's an ongoing work with a number of different collaborators. So these are the students that have been involved in the synthesis, and so key collaborators are Associate Professor Gedwi Chio-Wonkamon, Professor Subhanan Bua, Professor Simon Ward from Sussex, who helped with synthesis, and then funding providers. So with that, I'd just like to thank you for listening, and thanks to the committee for the kind invite, and if you've got any questions, I'd be happy to take them.